And welcome back to another educational video. Today's topic is cell and gene therapy, one of the most exciting and rapidly evolving field in the modern medicine. Over the past few decades, healthcare has made tremendous progress. We have developed advanced medicine, sophisticated surgical procedure, and innovative treatment strategies that have significantly improved patient outcomes. However, despite all this advancement, many diseases still do not have a permanent cure. What if we could repair damaged cell instead of simply treating the symptoms? And what we could replace a faulty gene responsible for a disease, or even engineer a patient's own immune cells to fight cancer with remarkable precision. This is exactly what cell and gene therapy aims to achieve. This revolutionary technology are transforming the treatment of cancer, inherited genetic disorder, rare diseases, and many chronic illness that were once considered difficult or impossible to treat. In this video, we will build a strong foundation by understanding the basic principle of cell and gene therapy. Whether you are a pharmacy student, life science researcher, healthcare professional, or simply interested in the cutting edge medical science, this video will help you understand this concept in a clear and structured way. So let's get started. Before diving into the topic, let's first understand what we'll learn in today's session. By the end of this video, you will be able to define cell therapy and gene therapy and understand how these two approaches differ from the conventional medicines. We'll also explore why these therapies are considered revolutionary, where they are currently being used in clinical practice, and how they are changing the future of the medicine. Finally, we'll discuss some recently approved therapies that have already demonstrated remarkable clinical success. With this concept in place, we will able to prepare for the advanced topic that we will cover in the upcoming videos of this series. Now let's begin by answering an important question. Why do we actually need cell and gene therapy? To understand the importance of cell and gene therapy, let's first think about how most conventional medicine work. Traditional medicine are extremely effective at managing diseases, but in many cases they primarily control the symptom rather than correcting the underlying cause. For example, medication used to treat diabetes help regulate the blood glucose level, but they cannot restore the damaged insulin producing cells in the pancreas. Similarly, drugs used to manage the hypertension effectively control the blood pressure, but they do not permanently reverse the physiological changes responsible for the disease. Even in cancer treatment, chemotherapy and radiation therapy can destroy rapidly dividing cancer cells, but they may also damage the healthy tissue, leading to the significant side effects. Now consider inherited disorders such as sickle cell disease, beta thalassemia, hemophilia, or muscular dystrophy. This condition arise because of the mutation in the specific genes. If root cause of this disease is a defective gene, simply controlling symptom is often not enough. Instead, we need therapies that directly target the underlying cause. This is where cell and gene therapy represent a major breakthrough. Rather than focusing only on symptom management, these therapies aim to repair damaged tissues, replace defective cells, or correct diseases causing genetic mutation. In other words, the goal shift from managing disease to potentially curing it. That is why cell and gene therapy is considered one of the most important advances in modern medicine. And with cell therapy is a treatment approach in which living cells are administered to a patient to repair, replace, regenerate, or modulate damaged tissues or organs. The key point to remember is that the living cells itself act as a therapeutic product. Depending on the treatment, the cell may be obtained from the patient, known as autologous therapy, or from the healthy donor, known as allogenic therapy. In many cases, the cells are expanded or genetically modified in a specialized laboratories before being infused into the patient. One of the most well-known examples is CAR T cell therapy. In this approach, T cells are collected from the patient and genetically engineered to express a special receptor called as chimeric antigen receptor or CAR. These modified cells are then multiplied in the laboratory and infused back to the patient's bloodstream. Once inside the body, they recognize and destroy the specific cancer cell with remarkable precision. This innovative approach has dramatically improved treatment outcomes for certain blood cancers and has opened new possibilities in the cancer immunotherapy. Beyond oncology, cell therapy is also being investigated for regenerative medicines, autoimmune disorders, cardiovascular diseases, neurological disorders, and tissue repair. As the research continue to advance, the potential application of cell therapy continue to expand. Cell therapy is a broad and rapidly evolving field with several different therapeutic approaches. Each type of cell therapy uses specific population of cell to achieve particular therapeutic objective. One of the oldest and most widely established approach is hematopoietic stem cell transplantation, which has been used successfully for decades to treat leukemia, lymphoma, multiple myeloma, and various inherited blood disorders. Another major breakthrough is CAR T cell therapy, where genetically engineered T cells are designed to identify and eliminate cancer cells with exceptional specificity. Researchers are also developing CAR NK cell therapy, which uses a natural killer cell instead of T cells. And these therapies have the potential to provide safer, more readily available, and off the shelf treatment options. Another promising area is mesenchymal stem cell therapy, commonly known as MSC therapy. This stem cell possesses regenerative, anti-inflammatory, and immunomodulatory properties, making them promising candidate for the treating orthopedic injuries, autoimmune diseases, cardiovascular disorder, and tissue regeneration. Dendritic cell therapy represents another innovative approach that stimulates the patient's immune system to recognize and attack cancer cell more effectively. Finally, TCR T cell therapy involve engineering T cell receptor to recognize intracellular tumor antigen that are difficult to target using conventional CAR T technology. Although these therapies differ in their mechanism, they all share a common goal to harness the remarkable potential of living cell to treat the diseases that were once considered untreatable. In the next section, we'll shift our focus from cell therapy to gene therapy, where instead of using cell as a medicine, scientists directly modify the patient's genetic material to correct the disease causing mutations. Now that we have understood cell therapy, let's move on another revolutionary approach known as gene therapy. Gene therapy is a treatment strategy that focuses on modifying the patient genetic material to prevent, treat, or potentially cure diseases. Unlike the conventional medicine, which mainly controls the symptom, gene therapy aims to address the underlying genetic cause of the disease. To understand this concept, let's briefly recall the central dogma of molecular biology. Our genes, which are made up of DNA, carry the instruction required to produce protein. This protein performs essential function in our body. If a gene is defective due to mutation, the corresponding protein may be absent, malfunctioning, or produced in an insufficient amount, leading to the disease. Gene therapy works by correcting these genetic abnormalities through different strategies. Scientists may introduce a healthy copy of gene, replace a defective gene, silence a harmful gene, or precisely edit the DNA sequence using advanced genome editing technology. The therapeutic genetic material is delivered into the target cells using either viral vector such as adeno-associated virus or lentivirus or non-viral delivery system like lipid nanoparticles. A simple way to remember the difference is this: in cell therapy, the living cells act as a medicine. In gene therapy, the therapeutic gene becomes the medicine. Both approaches share the same objective to treat the disease at their root cause rather than simply managing their symptoms. Gene therapy is not limited to a single technique. Scientists have developed several strategies depending on the disease mechanism and therapeutic goal. The first approach is gene addition, where an extra functional copy of gene is introduced into the patient cell. This strategy is particularly useful when the original gene is missing or non-functional. The second approach is gene replacement, in which a defective gene is replaced with a healthy one. This restores normal protein production and cellular function. Another radially or rapidly advancing strategy is gene editing, where the DNA sequence itself is modified. Technologies such as CRISPR-Cas9, base editing, and prime editing enable scientists to make the highly precise genetic changes, offering new possibility for treating the inherited diseases. In some cases, the goal is not to replace or edit a gene but to reduce its activity. This is also known as gene silencing, commonly achieved using RNA-based technology such as siRNA or antisense oligonucleotides. More recently, mRNA therapeutics have emerged as another novel platform. Instead of modifying the patient's DNA, mRNA molecules temporarily instruct cells to produce therapeutic protein. The widespread success of mRNA vaccine during COVID-19 pandemic demonstrated the enormous potential of this technology. Together, these approaches have significantly expanded the possibilities of precision medicine and continue to drive innovation in the field of cell and gene therapy. Now that we have explored both the cell therapy and gene therapy individually, let's compare them side by side. Although both therapies aim to treat diseases at their root cause, they differ fundamentally in their therapeutic approach. In cell therapy, living cells themselves serve as a therapeutic product. These cells may be stem cells, immune cell, or genetically engineered cells that are administered to the patient to restore the normal function or eliminate the disease cells. In contrast, gene therapy uses genetic material such as DNA or RNA to modify the function of the existing cell within the patient's body. Cell therapy primarily focuses on replacing, regenerating, or enhancing the cellular function. And gene therapy focuses on correcting or modifying the genetic instruction inside those cells. Another important difference is the method of administration. Many cell therapies involve collecting cells from the patient, modifying or expanding them in a laboratory, and then infusing them back into the patient. Gene therapy, however, may be performed either outside the body, known as ex vivo, or directly inside the body, known as in vivo. Despite these differences, both approaches represent a major advances in personalized medicine and have the potential to provide long-lasting and, in some cases, curative treatments. Rather than competing technology, cell therapy and gene therapy often complement each other. In fact, many advanced therapies, such as CAR T cell therapy, combine elements of both approaches by genetically modifying the living cells before administering them to the patient. One of the most important concepts in the gene therapy is understanding the difference between the ex vivo and in vivo approaches. Let's begin with the ex vivo gene therapy. In this approach, cells are firstly collected from the patient, and these cells are then transported to the specialized laboratory where the desired genetic modification is introduced using an appropriate delivery system. After confirming the successful modification and expanding the cells, if necessary, they are infused back into the patient. Because the genetic modification occur outside the body, scientists have greater control over the process. They can verify the quality, efficiency, and safety of the modified cells before they are returned to the patient. This makes ex vivo gene therapy particularly suitable for blood disorders and immune cell therapies, including CAR T cell therapy. Now, let's consider in vivo gene therapy. In this approach, the therapeutic gene is delivered directly into the patient's body without removing any cells. Viral vectors or non-viral delivery system transport the genetic material to the target tissue where it enters the cells and produces the desired therapeutic effect. In vivo gene therapy is especially useful for diseases affecting the organs that cannot be easily accessed or treated outside the body, such as liver, eye, skeletal muscle, or central nervous system. Both the approaches have their own advantages and limitations. The choice depends on the factors such as the disease being treated, the target tissue, and delivery method, overall the treatment strategy. Cell and gene therapy offers several remarkable advantages over the conventional treatment approaches. Perhaps the greatest advantage is their ability to address the underlying cause of disease rather than simply controlling the symptom. Many of these therapies have the potential to provide the long-lasting therapeutic benefits after a single treatment. This technology also enables the highly personalized treatment strategy, especially when therapies are developed using a patient's own cells. Such approaches have already transformed the treatment landscape for the rare genetic disorders and certain cancers. Or despite these exciting achievements, significant challenges remain. One of the biggest challenges is manufacturing. Producing cell and gene therapy products require highly specialized facility, strict quality control, and complex manufacturing processes. Another major challenge is cost. Many approved therapies currently cost hundreds of thousands or even millions of dollars, limiting access for many patients. Safety is another important consideration. Immune response, unintended genetic modification, and long-term monitoring remain critical aspects of clinical development. Finally, regulatory approval is more complex than for conventional medicines because these therapies involve living cells or genetic material, requiring extensive evaluation of quality, safety, and efficacy. Despite these challenges, rapid advances in biotechnology, manufacturing technology, and regulatory science continue to improve accessibility, safety, and effectiveness of cell and gene therapy. These innovations are expected to play an increasingly important role in the future of the precision medicines. Now that we have discussed the basic principle of cell and gene therapy, let's explore where these technology are making the greatest impact. One of the most successful application is cancer treatment. Therapies such as CAR T cell therapy have demonstrated remarkable success in treating the certain blood cancer, including leukemia, lymphoma, and multiple myeloma. By engineering a patient's own immune cell, these therapies enable the immune system to specifically recognize and destroy the cancer cells. Another major area is the treatment of inherited blood disorders, including the sickle cell disease, beta thalassemia. Recent advances in gene editing have made it possible to correct or compensate for the underlying genetic defect responsible for the diseases, offering patients the possibility of long term disease control or even a functional cure. Cell and gene therapy are also transforming the management of hemophilia. Instead of requiring the lifelong replacement therapy, patient may benefit from the treatment that restore the body's ability to produce the missing clotting factors. In ophthalmology, the gene therapy has shown promising results in treating the inherited retinal disorders, helping restore the vision in a patient who previously had very limited treatment options. Neuromuscular disorders such as muscular dystrophy are also benefiting from the recent breakthroughs in gene replacement therapies. Beyond this established application, researchers are actively investigating cell and gene therapy for cardiovascular diseases, autoimmune disorders, neurodegenerative diseases, diabetes, liver disorders, and many rare genetic conditions. As research continues, the number of diseases that can be treated using this technology is expected to increase significantly. The field of cell and gene therapy is evolving at an extraordinary pace. One of the most exciting development is in vivo gene editing, where genetic correction can be performed directly inside the patient's body without removing cells. This approach has the potential to simplify the treatment and make gene therapy more accessible. Researchers are also working on next generation genome editing technology, including base editing and prime editing, which allow highly precise genetic modification with fewer unintended changes. Another major focus is the development of the cell therapies. Unlike the current personalized therapies that require the manufacturing cells for each individual patient, off the shelf product could be produced in advance and made available immediately when needed. Artificial intelligence is also beginning to play an important role. AI is helping researchers design better vectors, optimize manufacturing process, predict therapeutic outcomes, and accelerate the drug discovery. Manufacturing technologies are becoming more automated, scalable, and cost effective, which may help to reduce the cost of these therapies in the future. As scientific knowledge continues to expand, cell and gene therapy is expected to become an integral part of the personalized medicine, offering a safer, more effective, and potentially curative treatment for a wide range of diseases. The future of the medicine is moving beyond symptoms management toward correcting disease as its biological source. and cell and gene therapy is leading this transformation. Let's summarize the key points from the today's session. Cell therapy uses living cell as a therapeutic agent to repair, replace, regenerate, or enhance normal biological function. Gene therapy focuses on modifying the patient's genetic material to correct disease-causing mutation or restore normal gene function. Unlike conventional medicine, which often controls system, these advances therapies target the underlying cause of disease. We also learned that gene therapy can be performed using ex vivo or in vivo approaches, depending on the disease and treatment strategy. This technology have already demonstrated remarkable success in treating cancer, inherited blood disorders, hemophilia, retinal diseases, and several rare genetic conditions. Although challenges such as manufacturing complexity, high cost, and long-term safety remain, rapidly scientific progress continues to expand the possibility of cell and gene therapy. Understanding this fundamental concept is essential because they form the foundation for many of the advanced topics we will discuss throughout this series. That brings us to the end of this today's introduction to cell and gene therapy. I hope this video has helped you to understand the basic concept and appreciate why this technology are considered one of the most important breakthroughs in the modern medicine. If you found this video informative, please consider supporting the channel by liking the video, sharing it with your classmates or colleagues, and subscribing to Only Pharmacy for more in-depth content on pharmaceutical sciences, biotechnology, drug development, and regulatory science. If you have any questions, suggestion, or topics you'd like to share with me or you'd like me to cover in the future videos, please leave them in the comment section below. I'll be happy to hear your thoughts. In the next video, we'll begin our detailed discussion on stem cell therapy, where we will explore stem cell biology, classification, sources, mechanism of action, manufacturing, clinical application, and currently approved therapies. Thank you for watching. I'll see you in the next video.